Hello, and we're going to do something a little different for this chapter 5. I'm going to divide the chapter into two sections, even though the book has it all combined, because I think it's important to pay attention to amino acids discretely. So we'll do a little lecture here, we'll call it chapter 5A, and then the next lecture, which I'll post shortly, will be chapter 5B, primary structure of proteins. So with that, let's go ahead and jump right into it. Some of these slides will take a bit, some of them will be quick. So what I wanted to do is just take a look at some of the questions that we might want to focus on, give everyone a little bit of focus when they're reading through the chapter or studying. What are some of the properties of amino acids? And what are their structures, and how these structures influence their properties? How are amino acids acidic and basic? And what kind of equilibria do we see because of these systems? What kind of reactions are typical for amino acids? What are their stereochemical properties? And then, to a lesser extent, what are their spectroscopic properties? How are they analyzed and separated? And like I said, those last 3 points are less significant for me as an instructor than the first 4. So what are the structural properties of amino acids? Amino acids all contain a central tetrahedral carbon, with the exception of glycine because it has 2 protons and therefore it's not chiral. And there isn't that much common diversity of amino acids. There's 20 common amino acids. That doesn't mean that there aren't lots of variations on those themes, but in terms of a majority of the amino acids, if we focus on those 20, we get a lot of bang for our buck. Now, amino acids can be joined through a chemical reaction, forming a bond called a peptide bond. And we're going to get to that in the last quarter of the chapter, or of this lecture. And this is how we get long polymers of amino acids, which lead to proteins. There are variations on amino acids which occur rarely. And some of the examples that we'll look at will be rare in the sense that you don't see many examples of those occurring, although in nature they occur relatively frequently. And we'll just dedicate a little bit of time to those, although in terms of where should you be putting most of your attention, once again, back to the commonalities between the 20 common amino acids. And then every now and then, there are variations of amino acids where you don't see them in proteins at all. For instance, neurotransmitters. But we'll have a little extra material on that as well. So, what's the structure that I'm referring to which makes an amino acid an amino acid? Well, every amino acid has a central carbon with 4 groups on it, and 3 of the groups are common. There's a proton, there's an amino group, and there's a carboxylate group, or carboxylic acid. And we refer to it as carboxylate in biochemistry because under biological conditions, a vast majority of the time, the carboxylic acid is deprotonated and is therefore the conjugate base version, the carboxylate. Now, what gives each amino acid its identity is the presence of the R group. So everything else is the same. You have your proton, your amino group, and your carboxylate, but the R group defines the unique chemistry of any particular amino acid. And often these are referred to in biochemistry as side chains, although R group is more typical of organic chemistry nomenclature. One of the properties of amino acids is that they can simultaneously, and in biological conditions do, carry both a cationic charge with our amino group and a anionic charge with our carboxylate. So these are zwitterions under biological conditions, a vast majority of the time. Now, we'll pay a little bit more attention to the peptide bond down the road in this lecture, but just to make it clear, the formation of peptide bond is a reaction between the carboxylate, which we have, I need to reselect every time, which we have here, and the amino group of another amino acid. And what happens is two of the protons from the amino group and one of the oxygens combine to form water as a leaving group. And the loss of water helps drive the reaction, forming a solid, stable bond, the peptide bond between the two residues. So there are 20 common amino acids, and there are two different methods of abbreviation which refer to these amino acids. There is a 3-letter abbreviation, and there's a 1-letter code abbreviation. If you are going to work with proteins in your research and you're looking at sequences within proteins, it's essential to know these abbreviations. Likewise, each R group has unique acid-base properties or does not have any acid-base properties, and you need to know where an amino acid falls in terms of its general category. And there are 4 typically ascribed categories. Now, not everybody agrees on which amino acids belong in which category. Most of them, there's agreement, but there's occasional disagreement. So, when in doubt, go with the book. And these 4 categories are nonpolar amino acids, which we have here. And these nonpolar amino acids are hydrophobic. They tend to be involved in the hydrophobic collapse of proteins during the initial folding processes as they form their higher levels of structure. We have the polar amino acids, which don't carry charges on the R group, meaning they don't have acid properties, but they are polar. A good example of this would be something with hydroxyl group on the R group. And you're going to see a lot of involvement of these along the surface of proteins, occasionally in the interior, but predominantly on the surface, involving hydrogen bonding and nucleophilic interactions along surfaces and with substrates. We also have acidic amino acids, and this is referring to the R group. We know that we have the carboxylic acid functionality, but specifically the R group of each individual amino acid can provide it with acidic properties. So for any amino acid which carries an extra acidic functionality on the R group, at biological conditions, pH 7, you're going to see the addition of a negative charge because the Zwitter ion balances out. The amino group carries a positive charge, the carboxylate carries a negative charge, and this causes a net neutrality. When you have an acidic amino acid with an extra acidic functionality, under biological conditions, we're going to be seeing a net negative charge. Likewise, we have basic amino acids, and for the same rationale, they'll carry a positive charge under biological conditions, pH 7. And there aren't that many of the acidic and basic amino acids, so we'll be able to focus on those pretty easily. So let's see which amino acids belong in which category. That we have the nonpolar amino acids. And nonpolar amino acids include leucine, proline, alanine, valine, methionine, tryptophan, phenylalanine, and isoleucine. If you take a little look at each of these R groups, for instance with leucine, our first in the top corner here, you can see that we just have an isobutyl group as an R group. And this is classic nonpolar functionality. We don't have the presence of any oxygens or nitrogens or lone pairs hanging out here. I mean, we do with the methionine, we have 2 lone pairs on a sulfur, but it's contained, it's not on the end. You can make an argument for that having a little bit of polarity, but overall, net methionine is often considered nonpolar. We also see some unique structures where in proline is going to have some unique properties when we get into higher levels of protein folding in terms of how it allows or disallows for certain types of structures. because the R group actually comes back and reacts with the amino group, forming this 5-membered ring. So, still an amino acid, but the R group does react with the amino group. Moving on to the polar uncharged category. We put glycine often in the polar uncharged category because it does have a net dipole, which is pretty pronounced. However, glycine's the oddball because the R group is simply a proton. This is why glycine is achiral. It's the only amino acid that we're going to cover that is achiral, because anything other than a proton attached to the central carbon will make for a chiral center. So it's a bit of an oddball, and sometimes people will stash it in the nonpolar category as well, and you can debate that to the end of time, but for the sake of this lecture we'll go with polar. Likewise, and more simply actually, we can see the presence of our hydroxyls. We have some ketones, we have some lone pairs on nitrogens over here, ketones, we have lone pairs on our sulfurs. We've got another phenyl group here. It's not a hydroxyl, it's a phenyl group because it's attached to our benzene ring. All of these are polar groups, therefore polar, but not acidic, therefore not charged under normal physiological conditions. Oh, to review, names, because we want to know all of these. Glycine, serine, spargenine, glutamine, threonine, cysteine, tyrosine. Basic amino acids. So we only have 3. That's a nice relief, hasn't it? We've got lysine, arginine, and histidine. And each of these contain a nitrogen which is positively charged under physiological conditions. Therefore, these amino acids carry an extra positive charge as part of their Zwitter ion complement and are basic. Acidic, even simpler. We only have 2 to worry about, and they have the part acid attached to the name, which is awfully convenient. So we have aspartic acid, and we've got glutamic acid. And here we've got them drawn with acidic protons in place. Under biological conditions, those protons will be off, and you'll have a carboxylate there. So, just to jump in front of the elephant in the room, everyone's wondering right now, how much of this do we need to know? So, what do we need to know? Moving backwards, you do need to know the functional groups of amino acids. You need to know which amino acids go into which category generally, so when you're seeing them appear in sequences of proteins, you know what the typical chemical property of that amino acid is. I am not strongly attached to knowing every abbreviation of every amino acid. Just know that it's not uncommon on standardized tests to see those pop up, particularly in questions pertaining to amino acid sequences within a protein. They're not going to necessarily spell it out for you. So it's not a bad idea, if you're intending to MCAT, to invest some actual time and know the 3-letter and 1-letter abbreviations associated with these amino acids. So there are also, in addition to those 20, numerous amino acids which occur infrequently in proteins in terms of the diversity of proteins in which they appear. The reason I have to specify that is because one of the examples here is collagen. And collagen is not a rare protein. However, it's very unusual to see hydroxylysine and hydroxyproline appearing in any protein other than collagen. It's a unique adaptation which leads to its unique structural properties. So in that sense, it's rare, although it occurs frequently, or there's a lot of it. And these are just a couple examples. We will pay more attention to hydroxylysine and hydroxyproline. GABA, epinephrine, histamine, and serotonin are very important neurotransmitters and hormones. And you also see a lot of phosphorylated amino acids as part of signaling. And if we get, probably next semester, into signaling cascades and phosphorylation of amino acids and signaling. Zooming in on those neurotransmitters, gamma-amino butyric acid, GABA, is GABA. This is based on a slight modification of glutamic acid. Epinephrine, or adrenaline, is a modification of tyrosine. Histamine, everyone's, or many of our nemeses during the birch pollen event during the summertime, is based on histidine. And serotonin, which helps us relax, is derived from tryptophan, hence the myth about turkeys making us sleepy. So, coming back to the chemistry a little bit now that we've reviewed the structure and categories of amino acids. Amino acids are weak polyprotic acids, and there's lots of ways biochemists look at this, but in any, let me grab a different color here. Generally, when we think about a weak acid in 100-level chemistry, you can say any generic weak acid, HA, is in equilibrium where it dissociates to the free proton and the conjugate base. Now, in any aqueous system, which this is showing here, the presence of water is going to scoop up that proton and form hydronium instantly. So it's more correct to show a weak acid in the presence of water is in equilibrium, and as the acid dissociates, it forms hydronium plus the conjugate base. Now, for a polyprotic system, we have multiple acids, one more acidic than the other. What we're seeing here above is closer to it, where you have a pair, and the charge here is to show the presence of that amino group, which is protonated in an amino acid. So it's a little bit of a specialization in an aqueous system. Is in equilibrium. I'm just going to reverse the order here. We're going to lose one of the protons. The loss of that one proton gives us a neutral charge. And we've got our hydronium. So even though we're probably more familiar with this, this is simply an adaptation of what we're used to seeing. So let me erase this really quickly. Although we may have an expectation when we're looking at a weak acid in equilibrium where it dissociates to the free proton and our conjugate base, and we can describe Ka as being the concentration of the free proton times concentration of the conjugate base over the concentration of the weak acid. And we have an expectation of seeing this. Seeing something like this is exactly the same thing, but viewed through a slightly different lens, polyprotic specifically, and in an aqueous system. Now that I've kind of cleared up some of that, the degree of dissociation of the weak acid depends on the pH, right? We talked about that previously, where if you put something that's a weak acid in a more acidic medium, the acidic medium will act as the acid, protonating the weak acid, preventing it from being quote-unquote acidic. if you put that weak acid in a medium which is basic, or more basic than the pKa of that weak acid, the medium will act as a base and take the proton from that acid. Or conversely, you can think of that acid then gets to act like an acid in the basic environment and donate its proton to the medium. And this is a good example of this working in a polyprotic system. Starting with glycine, which has a proton for an R group, so we don't have to pay attention to it for a little bit. Let's see. We have pH of 1, pH of 7, pH of 13. Now, the pKa of the carboxylate or carboxylic acid, when it's attached to an amino acid, is about 2. So in a pH of 1, we would expect it to be 90% protonated. And that's what we're seeing here in the simplification. As we're seeing a protonation because the medium is pH 1, the pKa is about 2, and that varies a little bit depending on what the R group is, but we can round. And likewise, because the pKa is going to be over 10 for our amino group, it will be nearly 100% protonated and therefore charged. Now, this is important because although under biological conditions pH 7, where it's a Zwitterion, here, under very acidic conditions, glycine is fully protonated and therefore is no longer a Zwitterion because the negative charge is gone. It's now a carboxylic acid, not a carboxylate, and we only have a cation. If we start to make the solution more basic, tend it towards neutrality, we get the Zwitterion, or we have a net neutrality. And that's because if you have a pKa of around 2 here, and a pKa of around 10 here, pH 7, you're going to see nearly 99.9% plus deprotonation of the acid because the acid can behave as an acid and donate its proton to the medium. Yet we're still substantially more acidic than the pKa of the base, and therefore we're charged at both positions. And if we make the solution even more basic, tend towards pH of about 13, you're going to see an anionic form where the medium is much more basic than the weak acid, therefore it will be deprotonated, which we see here, but we will also end up deprotonating the base, which makes it lose the cationic charge. So over the spectrum of a pH titration, you can go from cationic to Zwitterion to anionic. Oh, here we go. I've been approximating 10. This figure says 9. You'll see when you look at tables of actual pKs for any individual amino acid that you have some variation between these values. These are good ballparks. So what is the alpha? Well, alpha carboxyl alpha amino. Well, whenever you have your central carbon and you have your proton and you have your R group and you've got your carboxylate and you've got your amino group, this is your alpha carbon, and therefore the carboxylic acid which is attached to it is your alpha carboxyl group. The amino group is your alpha amino group, and we have to specialize in those or specify that because there will be basic amino acids which have other amino groups, and there will be acidic amino acids which have other carboxylates. And so when we refer to the alpha amino or alpha carboxyl groups, that refers to the ones attached to the central carbon at the chiral carbon and is ignoring the potential amino or carboxyl groups occurring in the R group. Now, some of you may be fresh enough on your OChem to rethink, well, pKa of 2, that seems awfully low for a carboxyl group. I remember acetic acid, which is our most basic carboxylic acid, having a pK of 4.76. So why the difference? Well, these two functionalities affect each other pretty strongly, and the presence of them next to each other on the same carbon actually alter the pKa's pretty substantially, making the acid more acidic and the base a little more basic. So don't fret, it's not a trick. You don't have to get an axe. So let's take a look at the titration of glycine, since we just kind of ran through it. You'll see that this is just another way to look at the multiple dissociations which take place, where we're losing a hydrogen here, and we're losing another hydrogen here. Or we could think of it gaining if we're going the other direction, going backwards. So losing this way, gaining this way. Now we can also identify isoelectric point. Isoelectric point is simply the halfway point between the two pKa's for the different dissociations, the pK1 and pK2 being the inflection point where the slope stops slowing down and starts speeding up. So it's really easy to identify visually, or you can do it mathematically with a derivative. The pK1, of course, is where we have that equivalent ratio of protonation and deprotonation for the acid. pK2 is our equal ratios of protonation and deprotonation for the base, and the isoelectric point is going to be where we have that nice zwitterion. Now, let's take a break from glycine, since it's a little boring sometimes. I mean, it's not totally boring because, well, we'll have all kinds of proteins that rely on glycine to provide some unique structures, collagen being one of them, actually. But let's look at some acidic and basic amino acids and look at how those titration curves vary compared to the glycine, because here we're bringing another acidic moiety and another basic moiety to the party. So, along with that, we bring another dissociation. And let me see if I have room to draw here. I don't think I can tuck it in. I think I can explain it off the figure. So we have a situation where we have glutamic acid, which will have a positive charge, be neutral, a negative charge, and two negative charges. The isoelectric point is still defined as the average between pK1 and pK2. If you were to try to sum all three, that wouldn't find the isoelectric point because we wouldn't identify the neutral point. You always need to take the isoelectric point from the average of where you have a +1 and a -1 charge in your system. Likewise, we see that here. If lysine with a 0 charge, you want to find that isoelectric point, it's going to be between pK2 and pK3 because that's where we have a +1 and -1 charge. So if you're asked a question to calculate the isoelectric point and you're given the pKa for all of the different functionality groups of an acidic or basic amino acid, you have to know which one to ignore. You have to be able to identify that in the case of the acidic amino acid, where you have the extra charge, needs to be ignored. Likewise, for the basic amino acid, you have to identify which pKa you need to toss out of the equation. And this tends to throw people a little bit. But if you just start at the beginning and use a little bit of rationale for protonation and deprotonation, you should be able to establish where your charges are and identify which ones to use. Now, this is a big caveat. What I just showed you in the previous slide, I'll even go back there, are these nice, neat titration curves with discrete isoelectric points. If you put all these amino acids together in polymers and have them fold in on each other, making proteins with secondary and tertiary structures and quaternary structures, you're going to get a very large difference in the side chain pKa values of those R groups, because the chemistry of how these R groups associate with one another three-dimensionally will change their relative acidity or alkalinity. So we can pay attention to these as just pure amino acids, but realize that how they behave in a protein can vary depending on their chemical environment surrounding them. So this is a really classic problem that you see in a lot of multiple choice exams for biochemistry. What is the pH of glutamic acid solution if the alpha carboxyl is 1/4 dissociated? And it's not really hard, but there's a little bit of a trick to it, so I wanted to walk you guys through it. The trick is, everyone's used to seeing, well, not everyone, but most of you are used to seeing the calculation as shown. pH equals pKa plus log of our conjugate base over our acid. Now, that's basically what we're doing here. We don't need to actually know what the true concentrations of both these are if we know the ratio, because that's really all we're looking at here is a ratio. So if it says 1/4 dissociated, you need to do two important steps here. Which version is dissociated? If we have any particular weak acid, this is the protonated version, this is the dissociated version. So if something's asking if it's 1/4 dissociated, that value goes on top. If it's associated, it's here. And sometimes they ask for 1/4 dissociated and or sometimes they ask for 1/4 associated. And you just have to pay attention to specifically which one they want. Now, the second trick to solving this kind of problem is to realize that if for the conjugate base we have 1/4 association, that means 1/4 of all of the species in this acid-base equilibrium are associated, or dissociated, and 3/4 are associated. What that means, if we just do a little bit of cancellation of our denominators, is that we have a 1-to-3 ratio. It's very common for people to hear, oh, it's 1/4 dissociated, so this is 0.25, and this is let's see, sorry, I can't even do it wrong. And they go 0.25 to 1, and they end up with 1/4, and they get the wrong answer. It's 0.25 of 1/4 because the total of this plus this is 1. So if you use up 1/4 in this, only 3/4 can exist here. So in the end, it's a 1-to-3 ratio when something's 1/4 dissociated. And that's why we can plug it in to the equation here. Now the other part of this is that you need to know what your pKa is. And that's relatively easy. It's glutamic acid. And for the alpha carbon, which has pKa of about 2, to be a quarter dissociated, we would be focusing on that pKa. Why that one alone? Well, the others are so distant from it that they're going to not affect the overall, and they're not asking about the overall pKa here. It's just asking about the degree of association or dissociation of the alpha carbonyl. Conversely, what if we have a basic amino acid in lysine, and we want to know about the degree of dissociation of the side chain amino group? Ooh. Well, then you have to go look up, this isn't the alpha amino group, it's the side chain amino group, which pKa are you dealing with? So instead of it being 9, the pKa would be 10.5. You'd have to go to a table for that. I don't expect you to know all of the pKa's of all the side chains, although you should know that this one is basic because it's a basic amino acid. So generalizably, know the chemical properties. Specifically, we'll have tables. The rationale is exactly the same. I'm not going to go entirely through it, but we can identify our pKa that's appropriate. We know that on top will be our conjugate base form. On the bottom we'll have the acid form. So in this particular case, if it's 3/4 dissociated, that's our conjugate. 1/4 associated, force cancel, and we have a final of 3:1. And then we can just plug it in, plug and chug, get our solution. And you can always just kind of double check and say, does this make sense? Would a pH of 11 lead to a predominantly dissociated amino group? And the answer is yes, because the solution is going to be more basic than the pKa, therefore it will behave as an acid beyond just a 50-50 ratio, donating its proton and predominating in the dissociated form, leading to a 3:1 ratio. So it makes sense. So some of the other types of chemistries that take place with amino acids that are worth quickly reviewing. The carboxyl groups tend to form amides and esters. Amino groups often form shift bases as well as amides. And we also have some unique side chain reactivities, the most important of which, well, likely the most important of which is cysteine, because cysteine residues can form disulfide bridges which covalently link distant cysteines very close to one another three-dimensionally, although in terms of where placement is in the long polymer of the protein can be quite distant. So it can really strongly dictate some of the conformation of the protein. Now, some of the chemistries of isolating amino acids is very tricky because we have a lot of common functionalities on each amino acid. It's very unusual to identify unique chemistries to a unique amino acid. So actually doing bench chemistry on proteins is quite tricky. So going back to our cysteine and the disulfide bridge, this is what it looks like. So this is a cysteine where we have our alpha carbon with our alpha carb. This is in part of the polymer. Our carboxylate, our amino group, our proton, and the R group for cysteine. And so we have our pair of lone pairs on the sulfurs, and what ends up happening is we're going to lose, let me change colors, lose the pair of protons. We will form chemical bond between the sulfurs, and that is a covalent bond linking the two residues in a disulfide bridge. And this is a strong redox reaction. So let's talk about stereochemistry of amino acids briefly. Like I said before, every amino acid other than glycine is chiral. That's also one of the gimme multiple choice questions you see on biochemistry exams. What is the only achiral amino acid? Glycine. Now, the nomenclature for defining the stereochemistry at the chiral carbon for amino acids is archaic, but we still use it because biochemists hold on to things. Scientists in general hold on to things. And we use a DL nomenclature. Now, there's two types of DL, so I'll clarify that on a different slide. But just know that big D, big L is what we're using, and that's based on the DL nomenclature established using glyceraldehyde as a model. Now, L-amino acids predominate in nature. It's really nice to see texts changing their language, because they used to say L-amino acids are the only amino acid that occurs in nature. It turns out that D-amino acids do occur naturally, but very rarely in comparison to L-amino acids. There's actually been some really elegant studies showing the appearance of D-amino acids during neurodevelopment and embryonic development. It's just very hard to analyze the difference between a D and L amino acid using a lot of the chemical technologies we had available to us historically. So we're just now starting to really probe the role of D-amino acids in our biochemistry. But for a vast majority of amino acids which occur naturally, they are amino acids. Now, there's no doubt that using RS nomenclature we learned in organic chemistry is vastly superior as a means of defining chirality, particularly since there's amino acids which have multiple chiral centers, and we would be able to unambiguously define what we're talking about if we were using something like that. However, we don't always get to choose the tools that we prefer because of the field that we're working in. So, once again, we're going to have to get comfortable with big D and big L. So, I mentioned the use of glyceraldehyde as a model. What does that mean? Well, what it means is during the early establishment of chirality, glyceraldehyde was one of the model compounds, and glyceraldehyde was arbitrarily assigned D and L in a 50-50 mixture. Now, that person got lucky, and big D, big L for glyceraldehyde corresponds to little d, little l, which stand for dextrorotary and levorotary, which are optical rotation terms for positive rotation and negative rotation. In glyceraldehyde, big D corresponds to little d and big L corresponds to little l. The guy got lucky. However, the use of big D and big L does not inherently mean dextrorotary or levorotary when applied to other compounds other than glyceraldehyde. So, you can see here, structure of glyceraldehyde. We've got our aldehyde, got our alcohol, got our other alcohol, and got a proton. So how does that correspond to an amino acid? Like this, oddly enough. So the amino group of the amino acid corresponds to the the carboxyl group corresponds to the aldehyde. The amino group corresponds to the hydroxyl, alpha hydroxyl. The R group corresponds to the alcohol, which is not alpha, and the proton is just a proton. And that's how we have our nomenclature for D and L amino acids in biochemistry. Once again, this does not mean plus or minus optical rotation. It does not mean dextrorotary, lever rotary. It just means that these structures, the L-amino acid, has an analogous structure to L-glyceraldehyde. Now, if you are asked to draw an L-amino acid or identify an L-amino acid, there's a really neat trick. You don't have to memorize the structure of glyceraldehyde. What you can do is just use this Korn mnemonic. Really slick mnemonic where if you put the proton upward facing towards you and you have your carboxylate, your amino group, and your R group, and you look like you're turning right with a steering wheel, going clockwise, you spell Korn. So, not too bad. Not too bad at all. That is also not an uncommon thing to be asked of during standardized exams. What is unusual in a standard exam is any discussion of prochirality, but I really like prochirality, so I'm going to throw it in there. I hope you guys saw something about prochirality in organic chemistry. Some textbooks use it, some textbooks don't use it, but it's really important biologically, because although chemically, if you put these in a test tube, they will not behave with chirality or uniqueness. Our enzymatic systems have the ability to identify prochiral centers and react with them uniquely to produce a specific chiral outcome. So, what is prochirality? It's when something doesn't have a chiral center, but if you were to replace either one of two protons, like either of these two protons, with a higher or lower priority substituent, it would become chiral. So, which you can define HA or HB as pro-R or pro-S. Also, another method of prochirality is when you have a planar system which can have a reaction where something's added from above or below. The addition of something to that planar system with three substituents will now create a tetrahedral chiral center, as long as whatever is added is different than the three substituents that are already existing. Therefore, there's unique chemistry to the face that you're adding. You have this top face and bottom face, and they will give you different chiral priorities. I'm not really concerned with you guys determining pro-R or pro-S or Re or C faces, but I want you to be aware of what prochirality is and how biological systems can identify it and produce unique chiral outcomes, which we cannot make in a laboratory without enzymes. Why does this matter so much? That's extremely important, actually, because when we do benchtop organic chemistry syntheses, we get what you guys learned last year are racemic mixtures, where we have 50-50 ratios of plus minus mixtures at these newly formed chiral centers. A good example is ibuprofen. Ibuprofen, when you make it benchtop, has a racemic mixture at that carbon that I just identified. So it's actually, one's active and one is non-active. Now, it was commonly assumed a long time ago that the inactive form could do no harm. It was just assumed. The problem was identified later with thalidomide. Thalidomide was synthesized to be a sedative, and it was given to a lot of pregnant women. Unfortunately, one version of thalidomide was a sedative, and the other version led to severe birth defects. So it wasn't active as a sedative, but it was active in other non-desirable ways. Although we do these things, making racemic mixtures all the time benchtop, biology doesn't do this. Biology loves control. It likes a vice grip control. Imagine the loss of energy or control with the loss of chiral control. So, enzymes bypass this. Transitioning into some of the spectroscopic properties of amino acids. All amino acids do have absorption in the IR. However, only the ones with conjugated systems, phenylalanine, tyrosine, and tryptophan, absorb in the UV. And we typically analyze concentrations of amino acids because of fairly regular distributions of these, dragging things all over the place, at about 280 nanometers. Now, one of my pet peeves, if you're dealing with the quantification of a specific protein and you use a different protein for your standard curve, which everybody does, usually BSA, the ratio of aromatic residues in your standard will be different, very likely, than the ratio of aromatic residues in your isolated protein. You probably are going to get close, but it's not the best way to quantitate a protein. Doesn't mean that everybody doesn't do it, but it's inherently flawed and slightly inaccurate. If you're doing total proteins within a system, usually it all averages out and you get a lot closer. But just know the system you're working with and its limitations. As NMR, nuclear magnetic resonance, becomes more powerful, we get a lot more use of it doing protein structure and amino acid chemistries within proteins. So this is an increasingly powerful technique. 2D techniques like ROSI is incredibly useful because ROSI is a through space correlation technique where we can see which residues are near each other in space. So even though they might be 50 residues apart by primary structure, when the protein folds in on itself, voila, we can get all that information about how the protein's interacting. And if we put something else in there and the protein changes a little bit, we can see what those changes are. This is a really, really powerful tool. However, it is only really appropriate for soluble proteins because we have a lot of proteins that don't exist as globular soluble proteins. They occur in membranes. Their structure is defined by the membrane. You take that out of the membrane, you put it in solution, it no longer behaves like it would in a biological system, and the information you get is not correct. It may just even crash out of solution. So if you have an insoluble protein or a membrane protein, NMR is not the best technique. We do a lot of chromatography. Amino acids, because they have unique R groups, are easy to separate using liquid chromatography. We can also use ion exchange chromatography for bulk separation of amino acids. This is the type of chromatography which separates based on a pH gradient, which charges or uncharges different residues, and that affects the type of migration through the column. So we have charge-based chromatography with our ion exchange, and we have our HPLC, which is just based on any particular compound's relative affinity for C18 attached to silica bead. So just polar-apolar interactions. Coming back to peptide bond formation. The peptide bond, which I showed before, is the bond formed between, and let's see here, our carboxylate and two of the protons on the amino group, and that gives this new dipeptide bond. This results in the release of water, 1 equivalent of water. And peptide bonds have a very interesting chemistry, because looking at this, thinking of this as an organic chemist, you would say, well, you know, I bet that this thing has free rotation because it's a single bond. But then if you dig a little bit deeper, you'll see that there's some unique chemistry to this. It is not simple single bond with free rotation. You almost always see this dipeptide bond forming in a trans configuration where the proton is on one side and the oxygen is on the other. And the reason for this is partially steric, but also because we have double bond character in the bond between the carbon and the nitrogen. So between that carbon and nitrogen, you have about 40% double bond character. And that means that the pi electrons are being shared in a resonance system. We also see that we have a partial negative charge on the oxygen, and we have a partial positive charge on the nitrogen. This would also indicate some degree of resonance stabilization. What's really, really important about this is that because it has significant double bond character, this bond does not have free rotation. So I'm going to erase that so that everyone doesn't think that it has free rotation, because double bonds are rigid. And this creates a rigid system across the carbon-nitrogen bond, which means that everything's attached to those carbon and nitrogens are in a plane. So, what does this look like and why does it occur? Well, as drawn here, we have a lone pair on that nitrogen. That lone pair occupies a vacant p orbital. This lone pair, as part of resonance stabilization, can form a double bond between carbon and nitrogen. This exceeds the octet on the carbon. So the doubly bound electrons between the carbon and oxygen have to go reside on the oxygen, in vacating its double bond and putting the lone pair into a vacant p orbital that's non-hybridized. What this looks like when this occurs is we now have a positive charge on the nitrogen and a negative charge on the oxygen, because the nitrogen lost its lone pair and the oxygen gained a lone pair. So this is why we have partial negative charge and a partial positive charge, because of this resonance system where we have partial charges and we have partial double bond character between the carbon and nitrogen. And that character leads to this plane to which I referred. Notice that the carbon, nitrogen, alpha carbon, oxygen, and hydrogen are all in a planar system, also drawn like this. So we actually only have rotation, rotating all kinds of things here, around pivot points. And everything between the pivot points is planar. And this actually is really important for reducing the degree of freedom of rotation in sequences of proteins, which we'll get to down the road here. So, we're almost done with this chapter. Oh, excuse me, 5A of this chapter. A little nomenclature. Peptides. Peptides are just short polymers of amino acids, because as you form these dipeptide bonds between amino acids, you're just getting a long polymeric chain with no branching. And every one of those amino acids is considered a residue. So if you have two residues, just two amino acids bound to each other, it's a dipeptide. The minute you have any bonding of amino acids, it's a peptide. If you have three, it's a tripeptide. If you have between 12 and 20, it's an oligo or oligopeptide. And if you have above 20, you can just refer to it as a polypeptide. proteins contain one or more polypeptide chains. Often proteins will have much more than one polypeptide chain. It varies significantly from protein to protein. If you have more than one peptide, you refer to that protein as a multimeric protein. If it's multiple chains of the same kind of chain, it's a homomultimer. If it contains two or more different chains, it's a heteromultimer. So, and sometimes you can get specifics. For instance, a hemoglobin is a heterotetramer, meaning that it has different chains and that it has 4 different subunits, with 2 of them being identical and 2 of them, well, with 2 different types of alpha and beta. And that is the end of this part of the chapter. I'm going to get this one posted for everybody, and then I'm going to delve into the primary and secondary structure of proteins.